The overall goal of this experiment is to identify useful molecular markers for sinonasal carcinomas and olfactory neuroblastomas in a simple and efficient way.
This method can help answer key questions in the field of olfactory neuroblastoma and sinonasal carcinoma, such as the identification of molecular markers to discriminate tumor subtype. The main advantage of this technique is that we can obtain results in less than 6 hours or within a day for the hospital report. Demonstrating the procedure of RNA extraction and real-time PCR will be Giorgia Millefanti, a PhD student from my lab, and demonstrating the procedure of immunohistochemistry will be Giovanni Micheloni, also a PhD student from my lab.
To begin the procedure, collect and divide all the human FFPE samples into different subgroups according to the WHO classification of head and neck tumors. Heat the sample slides in an oven at 60 degrees Celsius for 30 minutes. Next, rehydrate the 3 micrometer thick FFPE sections by briefly washing them in xylene for 10 minutes. Then wash the slides in 100%, 95%, 85%, and 75% alcohol serially for 5 minutes each wash. Rinse the slides for 5 minutes in distilled water. Subsequently, block the endogenous activity by placing the slides in 3% aqueous hydrogen peroxide for 12 minutes. Afterward, perform antigen retrieval by treating the slides with 10 millimolar citrate buffer and microwaving for 10